All right, let's start our session. Welcome to track 2 of the first day of LifeScience Open Space. My name is Tomasz Kościółek. I'm a group leader at SANO, Center for Computational Medicine. And this session is called Pioneering Health Solutions, Unlocking the Power of Biotech and HealthTech. And really this is what this is all about. This is what we as SANO are all about, fostering collaboration between different computational groups, collaboration between industry and academia, collaboration between different specializations, collaboration with translation to the clinic, and so on. So this is very much near and dear to what we as SANO, as an institution, are all about. And this is very much also near and dear to myself personally. So today's session, today's track 2 is going to be split into two parts. Until 3 o'clock, we have a series of mostly the presentations from different companies, with one presentation from the Jagiellonian University. And a lot of what we're going to be talking about today in this first part revolves around clinical development, revolves around the microbiome and unlocking the microbiome's potential for health. And in particular, and I think this kind of embodies the spirit of life science open space anyway, I'm actually involved with the majority of presenting groups or companies anyway. So, great. So this is very much in the spirit of what life science open space is about. So please, I hope that by the end of today or by the end of life science open space, you'll be also able to say that you are indeed collaborating or exchanging knowledge with today's presenters. And please, let's keep in mind that today is not a research conference, it's not a self-promotion conference, it's a collaboration conference. It's we're all about talking openly about challenges that we're facing. We're all about fostering collaboration. So please keep that in mind during your presentations. And since we're a little behind, I waited for a little bit. So without further ado, let me introduce the first speaker, the keynote speaker of this session, representing Ryvu Therapeutics, Kamil Sitarz, who is going to tell us about building project value through clinical development. And I would like to remind subsequent speakers to please come down here before your presentation starts, so during the previous presentation, so you can get mic'd up. So please, Kamil Sitarz, welcome.
Good, thank you. Okay, good afternoon, everyone. My name is Kamil Sitarz. I'm from Ryvu. I'm acting as a Chief Operating Officer and also quite heavily involved in clinical development. That I want to give you a little bit of insight how we see it at. Okay. How we see it and what do we use it for. Obviously, some of the slides that you'll be seeing, most of you are quite familiar with, but I want to give you a little bit of insight behind how the project value is created over time. Because obviously, as an industrial company that Ryvu is, behind all the mission that we have and that we care about the patients, you also see a bit of potential of how the value of the project, also from the commercial perspective, can be built over time. As a company, we focus on everything what starts from the discovery, so when we have a concept, until the stage that hopefully, at some stage, our drugs will be available at the market to the patients commercially. But this path is very long. It takes multiple years, and it costs a lot of money. and a lot of investments, because quite a lot of biotech companies cannot afford, because they don't have active streams of revenues that can support it. What I will be focusing on today will be this part that spans between IND and NDA, so basically between the stage that you have your preclinically validated compound that was assessed to be safe in non-human kind of testing and can enter for the first time evaluation in the true recipient of a drug, which is the patient. And over time, with phase 1 study, phase 2, then phase 3, sometimes phase 4, you build the value of the project, because what you do, you gain more information. And you gain it in the relevant model, which is the human setting. In phase 1, you would test safety, tolerability, so whether whatever you are developing is good enough to be dosed. But then in phase 2, that's the stage that you normally would look into the efficacy. So you have the right dose, the right way of kind of giving a compound to patients, and then those patients, they hopefully start responding. And what you also see from the slide, that the funnel of this drug discovery kind of narrows down at phase 2, because this is what is typically the major problem of drug development, that whatever you have established in your in vitro, in vivo model, no matter how many animal models were used, that's the phase 2 study. Sometimes in phase 1, you have some initial signs that tell you whether the human being is truly responding to your therapy. And this is what we are all aiming for. Then phase 3, when you can expand and hopefully at some stage get submission and be to the market. And in numbers, it also shows that in order for the drug discovery development company to end up with this one approved compound, which is available for the patients, you need to start a multiple program journey. And many of them, I would even risk saying that most of them will fail. And, but you still hope for this one. And the project value is built, but how to assess the value of those projects and hopefully how to maximize it, this is what I would like to tell you a little bit more in the subsequent slides. So how to assess project value? There are many ways of what you can say. So one could say, let's just see what happens, and if someone wants to buy it, that will be our value. But over the time, you need to invest in this project. You need to encourage others to either buy part of it, finance part of it. You need investors, you need partners. So, and even for yourself, you need to see whether it makes sense to have a therapy in a specific indication. So for this, you need to have a valuation method. There are a couple of them, which cash flows with NPV, so net present value. The one which is the most commonly used in the biotech industry, in the pharma industry, would be a specific version of an NPV assessment, which will be risk adjusted. And what this risk adjustment is, basically, it's telling you how likely it is that something will succeed or fail in subsequent stages of discovery at the beginning and later on in development process. And there is an equation that can be used for it. I won't be going into numbers, or maybe not going into numbers per se, but telling you which elements can be modified and how. Obviously not keeping in mind that the presentation is only for 20 minutes, probably we will not go into all the possible details. And I will try to give you also some cases from our own experience, how we approach it at Ryvu. The order is, at least in my opinion, important, and it's subjective. That's my way of thinking at what I would like to put first. So starting with the probability of success, how can we increase the probability that our drug development program will be successful, that all the investment we will do in clinical trials will make sense? So to do that, you need to have a strategy. And it's not just that, let's design a clinical trial and let's see what happens. It's a much broader thing. So you need to know which patient, which indication, how to dose, whether it makes sense to dose it kind of in a limited population or a broader one, what's the market, how to access it, what is your interface with the regulators. Because we need to understand that in the end, we can have a beautiful therapy, but someone needs to tell, yes, it's safe, yes, it's efficacious, we will introduce it to the market. And those guys are regulators, FDA in the US, EMA in Europe, and some others. From the cash flow perspective, it's relatively easy. You can either increase the revenue stream or you can optimize the cost. Both of them can be done at the same stage. And not all of them are purely related to savings, but I will talk about it in a moment. And obviously, in the end, when your therapy is in the market, you want to maintain it for as long as possible. And to do that, you need to think about your patenting strategy, so the way that you can ensure that your drug will be on the market with exclusivity for longer time well ahead. So you don't do it at the end, you do it from the very beginning, having hopefully an umbrella strategy to that, looking in different options what the patent can be secured for. And I will walk through a couple of elements and some examples from Ryvu. So probability of success, comprehensive development strategy. So one would say, let's have a clinical development plan. It's a medically/regulatory oriented plan for how to run clinical trials in phase 1, phase 2, phase 3 in order to reach the market. But this is not something what's kind of fixed. It needs to evolve over time. The market changes, your competitors are on the market. Also, your situation may be changing. So this is something what needs to be kind of adopted on the way. But even at the very beginning, you need to focus on a couple of elements. And the rule of right tissue, target, safety, patient, it's relevant. So you cannot be just assuming that everything what worked in vitro and something what worked in vivo will be good enough. Test your patient population. Is it feasible to find patients with your specific, genetic mutation that will be of interest for you? What about your competitors? Is the target exclusive enough? Aren't other clinical trials run right now that could use a different mode of action but specifically address the same patient population? So with all of that, you aim for having your therapy to the market as soon as possible. Obviously, one could say, let's do phase 1 easily, phase 2 easily, then there will be phase 3. The cost increases as time passes. And what we try to do at Ryvu and many other biotech companies, we are trying to find a way how we can get to the market sooner. So at the stage where you don't have to go do all those large clinical trials and invest tens of millions of euros, but you can access market because your therapy is specific, that it gets to the right patient population that is needing therapy because nothing else, for example, works. And this is when regulators can give you a breakthrough therapy designation. They can give you an accelerated approval pathway, allowing you to start selling your drug on the market. Obviously, still some development needs to be done in parallel, but you start generating revenues. And it's a win-win situation. Patients have access to your therapy. It works because you have confirmed that. And the company has a stream of revenue that can later on finance other programs. You have seen from the first slide that many programs have to be financed to be unsuccessful in order to have the drug on market. So with this, planning both the interactions on the scientific, medical level, but also with the regulators is of essence. And how we approach that with Ryvu. So Ryvu 120 is our main asset. That's the compound that was discovered and is developed by Ryvu solely. We have all the rights. That's the first-in-class CDK19 inhibitor. So the mode of action that is presented by Ryvu 120 molecule is specific, and we know it quite well. And what we did, we have created a broad clinical development plan that tests Ryvu 120 in multiple phase 2 studies. So basically, we have confirmed first the safety in phase 1 development, but later on, we have planned our approach with specific phase 2 studies, either as a monotherapy or in combination with other drugs, in order to get an accelerated approval. And this is something what's testing your hypothesis basically on the fly, but requires also a lot of attention, not only from the medical scientific environment, but also from the regulators, because just to remind you, someone needs to tell you that you are allowed to be on the market. So this is how we introduce that in our common practice at Ryvu. Also keeping in mind that the value of the program can also be built through parallel translational activities that later on can be moved into clinical development. But we go broad because each of those streams, even if it's not correlated in time, can generate a different stream of potential revenues and give some benefit to patients. And with the probability of success, also what's important, and I mentioned that this funnel of go, no-go decisions in order to move from one phase to the other one can be put into numbers. And those numbers show the probability that a specific indication, a specific therapy being in phase 1 can be transitioned to phase 2 to phase 3. And you can see that it's not that it's 99.9 or 99.8. It very much differs. Also, depending on the indication, some indications require specific formulation of your drug, the way how it's administered. Also, the kind of the basics for the disease are also not that well known, and you need to test it in life in your phase 2 study. So please look into phase 2 to phase 3. This is where truly this thing gets verified. So how to ensure that my phase 2 study may be a little bit more successful than the other? It's good to have some preliminary clinical proof of concept. So this is why in oncology drug development, it's kind of obligatory to start your phase 1 study already in the population of patients. So you don't do in healthy volunteers because it's unethical, because those patients, they don't have any other alternative, so you can start your phase 1 study already in a given population of patients. And this is exactly what we did with our phase 1 study. We tested originally Ryvu 120 in a population of hematological disorders. The disease is called acute myeloid leukemia, but there is also an associated disease called high-risk MDS, so high-risk myelodysplastic syndrome that leads to AML, so to blood cancer, basically. And in our phase 1 study, we have seen a lot of patients with some sort of benefit. So you may look for a complete response of the patients, but you also should be looking into other ways that patients are responding. And in our phase 1 study, out of 30 patients, 15 of them responded somehow. So it was either a complete response that we have seen, either responses on the level of different blood cells. And with all of that, and also a bit of preclinical development we had underneath, we came up with our hypothesis of what will be the most beneficial, the population that will benefit the most likely in phase 2 study. And this is what we put at the beginning of our phase 2 development for Ryvu 120. So having clinical proof of concept really helps. But then the question is, should you be first in class or best in class? So 3 minutes. Okay. So with this, obviously being first in class is great, because that means that you know something the best. But being best in class means that you do it in the best possible way. And please remember, in the end, there will be doctors that will be selling it, and they get used to certain therapies. So even if you have something what's not first in class, but best in class, it's still worth to be considered. Your patient population that can bring up the market, and you can build your story around the whole pipeline in a pill. And optimizing, I will have to skip through that. I'm sorry. This is all very interesting. I talk too much, definitely. But this is all relevant. Maybe one thing to focus on. When you do your clinical development, don't think that someone will manage it for you. Manage it yourself, and do pay attention to it. Do it in a smart way. And make sure that your IP protection is in place from the very beginning, because this is what's building the value of your program. And that's the last slide. This is to show that those valuations, they are not only needed for yourself or just to give an equation. Analysts from banks that later on invest in your company, if you're a public company, would use RNPV method to calculate the value of your program, and they will adjust it as you proceed. So the data later on is moved into changes into your R in the model, and this builds the value of the program. So all those elements are relevant. Please use it, and hopefully that will help to build the value of your program, not only in early stage development, but also late stage, to give the benefit for the patient. So with this, I would like to thank you.
Fantastic. Kamil, do you have any calls for collaboration or open kind of challenges to the community that you would like to highlight?
We have a lot of challenges ourselves right now with the clinical. I think one thing that I see as someone working for a couple of years right now in clinical development, we really need to ensure that clinical trials as a way of removing R and helping the drugs to be in market are less. So they need to be regulated, but they need to be done in a common sense approach. So the sooner we can test in patients, the sooner the therapy is available. And we really talk about patients that are in need. So it's not business, it's the sick patient which is at the end. So let's work on it. And I can only tell that in Poland, there are quite a lot of initiatives around Medical Research Agency, ABM, Agencja Badań Medycznych, that really can help in the future, hopefully, to also organize clinical trials and make them smoother.
Fantastic. Thank you very much.
Thank you.
All right. Thank you. Thank you, Kamil Sitarz. That was an excellent keynote and an excellent introduction of you, like an excellent perspective from an established company in the more of the traditional pharma space. And now we have a triad of presentations revolving around microbiome, revolving around this new frontier that gives a lot of potential and hope for new therapies, new approaches to human health. So we're going to hear from, we're going to learn about how to better understand the microbiome to make it better work for us. But then we're going to also hear about how to develop microbiome-based biotherapeutics. And finally, we're going to hear from Agata Szymanek in this triad of presentations about the microbiome, about how different technologies that can help us also to characterize the microbiome better, faster, and cheaper. So, without further ado, let me introduce Paweł Łabaj, the CEO, co-CEO, the other one is me, of One Biome limited liability company. And Paweł Łabaj is also the head of the Bioinformatics Research Group at Małopolska Center of Biotechnology, as well as a deputy director of Małopolska Center of Biotechnology. Paweł, the floor is yours.
Now it's working. Okay, so thank you, Tomek, for a nice introduction. So now you know how to be the first speaker after the keynote. Okay, so I'm going to talk about how to get to know your microbiome, and one of the microbiome, because you probably have heard that in principle, we have many microbiomes on us, in us, around us. And to some extent, you can think about them that they are something like a force field, which is protecting you from the bad influence of the environment in some cases. But today, I would like to talk about the gut microbiome. So what we have in our guts and how it's affecting our health and well-being. Because when you will look back for maybe last 100 years, maybe a bit less, we have a huge civilization progress. Right? We have a lot of progress in terms of the technology. Unfortunately, it's also causing the environmental degradation. And this technological progress is giving us a lot of opportunities. We can explore more. We can get to know more. We can sense more. But everything makes that we are simply speeding up our life. Some people say that the evolution of our body, it's not catching up how fast we are speeding up our life. So it's nice. We can do way more than our parents, grandparents, great-grandparents, but it is affecting how we feel and how we will feel in the future. So our life comfort can already decrease with all these things, speeding up the life, bigger stress, also the degradation environment. But it can be also that our life comfort will decrease in the future because we do not take care and protect at the moment. And the interesting thing is that with the gut microbiome, we can see this there in the composition, in the functions which we observe. Because the gut microbiome is some kind of the mirror of our health status, of our well-being. So when we see that something is not going on properly, and if it's not very radical, when it's not really needed very severe treatment like Jarek will be talking in the next talk, we can think about whether the change of our lifestyle, maybe small modifications, small interventions might help. Of course, we are working, we are aware about global warming, we are trying to make some progress in it, so we shape our environment and in turn it influence on us, but it's not that fast progress as we would like to have. So what we can else do, we can think about whether we can adjust and change our diet and lifestyle, and by this influence how our microbiome will look like, and by this we can influence how it will provide maybe better well-being for us. So what we do in One Biome, we provide such kind of the care for our clients. So you can order a set, the collection set, then take a stool sample, send it to us. We will use our model to predict what kind of the changes might be useful. It will be consulted with our specialists, and then the specialist will take care of you. But then you can think about, okay, I think I've seen some advertisement of other services when some kind of microbes, they were assessed. So how you really differ? So what is the unique of our product? And here, the thing which makes us unique is also the entry point for the potential collaboration with maybe many of you. So both me and Tomek, we are coming from the science, from the research, and we have, each of us has over a decade of research projects, activities, where we're looking into research reproducibility, algorithm development, interpretation of the data, looking from the environmental perspective, gut microbiome perspective, development of the new models, and development of AI. And we see that this is really, especially when you look at the gut microbiome, it's really a complex challenge. It's not that you have specific profile and then you have a solution. It's not like that there is a reference genome and then I'm almost identical to this reference genomes. It's not like this with the gut microbiome. So for two different persons with exactly the same microbiome profile, the solution might be different because it also matters everything what is going around. So what we're specifically looking at is that we know that the technology can be a limitation. That's why we are going for the best available technology for measurement. We are using the third generation of long-read sequencing. We know and see that AI can help us a lot. It will not replace us, hopefully for a long time, but at the moment it can help us a lot. In this case, it can aggregate the whole knowledge about the gut microbiome and summarize what we should take into consideration when we're thinking about the modification of single-person diet and the lifestyle. And I think what is the most important here is that we provide the care by experienced specialists like dietitians. Because these people with huge knowledge and expertise can adjust what we'll get from AI to the specific person. And these people can take this person by hand over the time. Because the last thing which is unique for us is that we do our analysis of changes over time. Just one time point, it's not enough. You need to look how it's changing over time, how it's reacting to interventions, how it's reacting to the changes of the lifestyle, and whether it's really converging to the direction we would like to go. Without this, we cannot really say that we are affecting and improving the gut microbiome. And this is the place where we are really seeking for collaboration. Because all of this needs to be taken into account and analyzed in the context of activity and the feedback from our client. Of course, at the moment, this is more like that this person say, okay, I feel better, but it could be also a placebo effect. We are looking for other ways of measuring the whole exposome, so all these exposures factors which are affecting the specific person, to be able to trace whether our suggested changes in the lifestyle and in the diet are really improving some specific factors which we can really measure. Because it's not just that this is just we are here. As I have said at the beginning, the microbiome is something like the force field. And we need to look into the interaction between all this environment, all the exposome, with our body, and to be able to assess whether this interaction is going to the right direction or not. We are talking here about the wearables just to measure whether our general, let's say, parameters becomes better. We can say about some kind of the assessment of how fast we can learn. So in all these areas, we can seek for the collaboration, because the more source of the data we can put as a feedback into our model, the better recommendations for in the future products we can provide. So this is like last slide. Our core team at the moment is Tomek, who introduced himself already. I'm here. I'm Paweł. There's somewhere here who is also Michał. Tomorrow will be also Kamila. We have also more people collaborating with us. And if you'd like to get in touch and think about possible collaboration, maybe you are a specialist, maybe you have a network of specialists, maybe you are providing or developing the mobile apps for the wearables, we are happy to talk about the possible new products where we can integrate our expertise, your expertise, just to look for something new. You can find us on the LifeScience Open Space portal. And I also will be here the whole day, today and tomorrow. So just don't hesitate and just pop by. Thank you.
Fantastic. Thank you very much, Paweł. Pass this on to Jarek. Fantastic. So we heard about how the microbiome may be used for wellness and well-being. And now also from a collaborator, Jarek Biliński, we're going to of the Human Biome Institute. We're going to hear about how the microbiome can be used as a biotherapeutic. Jarek, the floor is yours. Please remember, talk about challenges and collaboration. Thanks.
Of course. Thank you, Tomek, for the invitation. It's great to be here. Hello, everyone. So yes, I will be talking about challenges and collaboration proposal for the community. We work as a company in the therapeutic area, so maybe just one sentence to add for what Paweł said, because he mentioned about microbiome itself and how it can change. So after all of the research right now, it is well known and quite well proved that microbiome disruption is the X factor in the most disease pathology. So when the diseases was called idiopathic, so there was no clue what is the real cause of the disease, it came out that mostly microbiome could be involved in the disease pathophysiology. And we as a company work mostly in the immunology part of the microbiome as it exerts a role to the human body. So in immunology, cancer, autoimmunity, and antibiotic resistance. So the company was founded 5 years ago, and it was based on my previous research in Medical University of Warsaw. The first, what we proven is that microbiome could be colonized with antibiotic-resistant bacteria. It's a marker of gut dysbiosis, and we found that because as I am a hematologist, a physician, I used to work in bone marrow transplant unit. We found that if the patient is colonized with antibiotic-resistant bacteria, so the patient has microbiome disruption, the 2 years overall survival, Okay, it's working like this. After 2 years after bone marrow transplantation, non-colonized patient was alive in 74% and colonized one in only 34%. And this death was not due to cancer, hematological, mostly cancer relapse, but non-relapse mortality, which means infections and immune complications. And this was the first time when we shown that microbiome disruption could cause the increase the mortality in those patients. So the next step was how to change these fatal outcomes. And the biggest intervention to change our microbiome is to replace the microbiome to a healthy one. So we did a so-called fecal microbiome transplants, and it did work. 60 to 79% of patients got rid of antibiotic-resistant bacteria from the GI tract after a month of therapy. When we found more, even if the ARB was not decolonized, the number of infections decreased of 49% in 6 months after fecal microbiome transplant. So we found that it is not only a ecological niche and antibiotic resistance decolonization module. It is also like infections decrease properties. So what are the challenges? The keyword right now. So today's standards to replace the microbiome to a healthy one is fecal microbiome transplant. Of course, we can use the diet, the probiotics, and so on, but the most radical method is to use microbiome from the donor, who is very healthy, and to introduce it to the patient's gut. This is very primitive, like you hear it. No matter that we are doing it very carefully, we screen the donors. I think our products are really one of the best in the world, are very safe and efficacious and potent, and it really works. We have a 90% of first indication cure C. diff infections, but the future belongs to donor-independent biosynthetic microbiome, biomimic one. So rebuilding the full ecosystem of the microbiome, but not using a donor, but generating the potential, the healthy microbiome as a set of genes and some factors biomimicking the full spectrum of the microbiota and fully synthesized in the bioreactors. So to do this, we've created deep tech laboratory and computational platform composed of 4 divisions, 4 steps, as we call it. The step 1 is proof of concept, when we use the donor-dependent microbiome to check if it's in some condition it's work or not. So we have some clinical programs looking for microbiome usefulness in those diseases. The step 2 is biobanking. So we have all changed to biobank the samples from patients, from the donors, before and after therapy. We have already over 20,000 biobank samples from over 800 individual patients. And we use those samples to look deeply into the microbiome, what is really working in this therapy. So we've composed a computational platform. It is called Trap Microbes, like the Trap 4 microbes as itself, to predict what the composition of the drug should be to cure the disease we are working with. So we are dealing with. So the platform itself is looking what particular genes, particular metabolic pathways, particular phages, particular bacteria are involved in the disease treatment. And the step 4 is synthesis of the drug, and this is right now what we are facing. So we have those 3 steps in our lead program done, and right now we want to synthesize the first dose of the drug. And this is the platform, and when we can say that our mission is to develop, we call it internally new generation biotherapeutics, enabling cure of any microbiome-related disease in a smart, AI-guided way, because we predict what the composition of the full microbiome should be to treat the disease. And this is also the space when we can collaborate. So right now we are looking for wet lab specialists, especially microbiologists, molecular microbiologists. If you are good in bioreactors, anaerobic condition chambers, drug synthesis, and all of this, let's say biotech specialists directed toward culturing cells, even if you have experience in human cells, we have just starting the program to build our own deep tech laboratory, fully anaerobic. We have a great team of people, of specialists, group leaders, but of course we need more. And our lead program, as of course we can work on any indication, and we have many clinical programs. Our lead program is to decolonize antibiotic-resistant bacteria and prevent the bacterial infections. We want to produce a immunobooster therapeutic, which is to diminish the graft versus host disease complication after bone marrow transplantation, and to immunomodulate the immune system to better treat cancer, because this is what we are right now developing. And in the space of longevity well-being, to optimize our microbiome, to have properties to live longer and better. And why the lead program is ARB? Because the market is one of the biggest in the world right now in the therapeutics area. Imagine that almost 1 billion people in the world is already colonized with at least one clinically relevant antibiotic-resistant bacteria. And half of the oncologists in the UK are worried that the patients will die of the infection after chemotherapy, not of the cancer itself. So this is why this program is the first one. We open totally new niche as a prophylaxis against invasive bacterial infections. So one step is, one cluster could be vaccines. The first vaccine against antibiotic-resistant bacteria is in development. The first one is in phase 2, but we are covering much broader space. So this will be the first prophylaxis, and you can think of it like an oral super broad spectrum, super powered vaccine with totally new mechanisms. And this is why we call it Microbiome 3.0 approach, that we are trying to do fully biosynthetic and intelligent microbiome drugs. So we need you, if you are interested in the computational space and mostly in wet lab space, to collaborate. And thank you. You can reach me through the app.
Fantastic. Thank you so much, Jarek. Thank you so much. Thank you so much, Jarek. And to close off the triad of our microbiome-focused presentations, we're going to hear about technologies, this time a little bit more, from Agata Szymanek from Perseus Biomics. Yeah, I didn't pronounce it wrong. Also one of the collaborators of us and Sano. Agata, the floor is yours.
Thank you.
Please
Yeah, all the roads lead to Sano, no, after all. Okay, so we said a lot today about the importance of microbiome for our well-being. We also said that it can actually become a treatment to deal with a lot of challenges that we have in our healthcare. And I would like to tell you why the microbiome research is facing bottlenecks in terms of taxonomy profiling of the microbiome. And what we would like to do in order to remedy the problem. So currently, microbiome we're trying to apply in multiple areas, including precision diagnostics, development of therapeutics, as Jarosław already said, but also personalized nutrition, as Paweł discussed. And there's plenty of other research in which microbiome is one of the key players. But majority of the insights that we have right now in metagenomics comes from the sequencing-based experiments. And they are facing significant difficulties in terms of exactly quantifying the proportions of the bacteria that we can find in the samples, simply because of the way the technology operates. This is, of course, impacting the precision of the insights and sometimes also the reproducibility of the experiments. The resolution of the taxonomy that we are able to provide is also limited, so we are not always able to point to a specific bacterial strains that is causing a specific issue. But most importantly, the research is progressing slowly because the time needed from sampling to getting the insights is really long. We're talking here about weeks from the initial experiment to a complete taxonomic profile. And of course, this is impacting all decision-making process in the R&D, but also it means that the insights for your patients, consumers, and for your business are simply a surface level. At Perseus Biomics, we are working on technology that we call DynaMap, which stands for Dynamic Microbiome Abundance Profiling. And ultimately, we want to be able to enable the laboratories to profile complete microbiome sample within 6 hours. Additional benefit of using DynaMap will be possibility to resolve individual strains in the sample, reduce the bias in the quantification of the species that you find there, but also allow you to profile your sample much cheaper than sequencing-based solutions. How we do it? We are leveraging technology of optical mapping for DNA, which is a really interesting concept because we don't really need to know the exact sequence of the bacterial genome in order to decide who is present in our sample. The way optical mapping works is that we know that certain specific, very short sequences are occurring in the bacterial genomes regularly and are spaced with different frequency alongside different genomes. By extracting very long DNA fragments and labeling fluorescently those sites, we are able to precisely point from which genome the DNA fragment was coming from. So what you can see here is the microscopy image of such a microbiome sample, from which we take back the signal, refer to a reference genome, and establish the bacterial profile. And such a scan takes around 20 minutes itself. If you want to know more, you can have a look at the mode of action video. It's visualizing pretty nicely of how the whole process works. So the goal here is, first of all, to provide you the service of microbiome profiling, and this is already possible with 5 days turnaround time. You can send your sample to our central lab in Leuven in Belgium. But our goal is also to establish satellite service labs all across the Europe and beyond to disseminate the technology in new geographies. So you could become one of our satellite labs if you have a core facility or contract research organization or you collaborate with one. That's one of the options to work together. But at the end of the first quarter next year, we will also start disseminating benchtop devices that will allow you to profile microbial sample within 6 hours in your own lab. So just to show you where we are with the technology development, we compare ourselves mainly to shotgun metagenomic sequencing because both are based on full microbial genomes. This is an example of 8 different fecal samples taken, and you can see that the correlation of optical mapping profiles and shotgun-derived profiles is actually really high. We also check how stable the method is, of course, because we want the insights to really be reproducible, to fight the challenge of reproducibility that other methods are facing. So what you can see here is that we took samples from different patients in biological and technical replicates. So biological replicates means we have the same stool donation. We take 2 separate samples, 2 separate DNA extractions, 2 separate optical mapping profiles. Technical replicates, we took 4 DNA. We took the same DNA from the sample 4 times, profiled it with optical mapping, and of course we calculated the distances between the profiles. And what you can see here is that the biological paired samples are grouping closely together, and technical replicates are almost identical in terms of their profiles, which shows high reproducibility. And also you can see here an experiment in which we took individual bacterial isolates and we profiled them separately against the reference database of all possible 15 strains. These are highly similar. We're talking here about the level of similarity of the full genome of above 99.9% in some cases. And what you see on the right-hand side heat map is that we are correctly identifying the strain that was present in the sample, even if we match against very closely related neighbors. And even if we are facing the clusters of sequences that are indeed almost 99.98% similar, the correct strain always comes up on the top, even if some cross-matches are occurring. And when it comes to the time of the service, that's how Dynamap compares to currently available solutions. So normally it's at least 10 days that you need to get the insights from the sample into the profiles. For us, it's going to be hours. And how can we work together? So right now, we are focusing on a lot of pilot projects that we would like to publish with our partners to build, of course, scientific trust in the technology. If you would want to get involved and become one of our research partners, then that's an option. We also apply for funding in Poland, which is what we did together, actually, with a research advisory support of SANO and Małopolska Center of Biotechnology. We're involved in a big international consortium studying multi-omics markers for optimizing antidepressant efficacy. So applying for international funding is also an option together. And we're trying to set up experiments where we could validate Dynamap against other long fragments technologies like Oxford nanopore sequencing, but also try and build good reference genome sequences, which we can see are improving the efficiency of the technology in profiling and quantifying. And bigger challenges that we're trying to resolve is how can we extend the technology to be able to profile not only taxonomy of the bacteria, but also their functionalities, and how we could use that to establish whether we are facing some antimicrobial resistance mechanisms in the bacteria. And of course, as I mentioned, benchtop device or satellite lab are pretty soon going to be available options. So on my side, if you would like to discuss the science behind it, then I will be somewhere around, and feel free to ping me up. If you want to go straight ahead to discussing the pricing or the details of the collaboration, then my colleague Melissa is the best person to reach out to. You can contact her via email. And of course, you can try to contact me through the LSOS platform as well. Thank you.
Fantastic. Thank you so much, Agata. Fantastic. Everyone's right on time, which is great. This is for you. This is for you. So continuing on, now moving away from the microbiome a little bit to other challenges, we're going to hear from Krzysztof Rataj of Ardigen, also a Kraków company, and also a company that exists within the SANO, greater SANO verse. So very much welcome participation, and we're going to hear about phenotypic screening for small molecule drug discovery. So going back to the drug discovery topics, the floor is yours, Krzysztof. And please remember to talk about challenges and collaboration.
Yeah, great. I'll be breaking a mold a bit here. So first, a bit of a pitch for the company. So we have customers all over the world, and we have over 180 people on board, with a quarter of them holding a PhD degree. So we have both technicians and scientists on board. We have a very broad spectrum of fields that we are engaged in life sciences, but what I will be more focusing on today is the phenomics platform and the applications in high content screening. So first of all, what is high-content screening? High-content screening is a method of visualizing cells and effects of the various treatments in cells via microscopy, where instead of getting a numerical readout, we get an image that we have to further analyze. First one was introduced in 1999 for transfluorescence assay for GPCRs, but since then many more came up, and these are usually targeted treatments where we have to do some kind of immunostaining or we have to have modified proteins that also have fluorophore attached to it, so that we can actually measure what we want our effect on the cell. So what was recently developed is the cell painting assay, where instead of targeting a particular protein or a particular set of proteins and seeing the effect of the treatments, what we can do is we stain various compartments. So in this case, we have nucleus, we have endoplasmic reticulum, we have RNA, mitochondria, and actin skeleton, and we'll get this very complex picture of the entire morphology of the cell, which has to be further analyzed. It's very easy, as it requires no specific cell lines or any modifications to cell lines, just simple fixed cell staining. It's cheap, and it allows for high throughput. So what's the usual process here? We first get some kind of, we have a cell culture, we perturb them with their chemical or genetic perturbations, so for example CRISPR or open ring frames inclusions. We can then perform the experiment, perform microscopy. We get then the huge number of data for cells, and this is the first challenge. From a single, let's say, middle range, high content, high throughput screen this way, we can get like 100 terabytes of image data. And how to analyze it in any way? Most of these images are, in the images between the negative control and the treatment are basically indistinguishable for even trained molecular biologists. So we have to apply machine learning and AI approaches to actually analyze the data. At our company, we developed a platform for this. We have a various set of applications from predictive, like mode of action prediction, biological property prediction, hit identification, and visual screening, through generative components where we generate new molecules that are likely to give a given phenotypic effect, together with custom analysis custom analysis pipelines for extruding as much data and information from just given treatment as possible. So first, I will highlight two of the recent projects that are quite interesting. And the first one was done in collaboration actually with Ryvu, where they had a co-culture of fibroblasts and a cancer cell line, a quickly dividing cancer cell line, and they wanted to find a drug that will kill the cancer cell line and not kill the underlying fibroblasts. So in this case, they performed the EDU staining. In this case, it targets quickly dividing cells, proliferating cells. So we have two different stains. We have the DAPI staining, so all nuclei, and the EDU, or here it's called FITSI, staining, where we only stain the proliferating cells. This is a very costly approach and a time-consuming one. So what we wanted to do is to whether we can actually predict the proliferating cell staining from the whole nucleus staining. We used a UNet model and trained it to predict, as we have as an input, the DAPI image. And then what we predicted is the mask for DAPI staining and prediction for the EDU or FITSI staining here. And as you can see on the right, this is highly correlated to the actual EDU staining. So when we then take the area that is predicted by our model that is covers the proliferating cells, it's very highly correlated to the actual number of of cancer nuclei on the given image. And based on this, we can predict even those response curves using just this kind of imaging. And our current study on the number of data points required to train the model, we can do it with as much as a single plate or even half of a plate of controls with both stainings to train the model for the entire cell line. It's very quick, so not even an entire day of training the model, and we get a twofold reduction in the cost required to perform a given experiment. So this was quite a success. We also have another case of hit identification for high content screening. So when we have a high throughput context of our experiments, so instead of doing a one plate or two plates, we have hundreds of plates with various treatments. We want to find out which actually wells represent a changing morphology of the cell. So what we devised is a model that you can feed with the negative control and the positive control data, and then it will predict only those wells which are very similar to the phenotype of the positive control and as far away from the negative control. Or we can do it in, let's say, any type of change approach, where we find all the wells where the effect is visible at all, anything diverging from the negative control. So what's the exemplary result here? So this is a, let's say, very good example of how a plate should look like. We have on the left side and the right side, we have negative controls, so these are not scored, so we are not interested in those. But in the middle, we can see the anomaly score for given treatments. And in this case, we have replicates in the top half and the bottom half of the plate. And we can see that we have, with the increasing concentration towards the top, so we can see that the anomaly score is rising with the concentration, and it's replicated between the replicates here. So it's a very nice image, and we can then see that these are the given treatments that we actually want to look at, because they have a high effect on the cell, and it's not arbitrary, and it's not something like an artifact, so we can work with this. But we also, what we found during this project is that our, let's say, hit identification approach can be also used to perform quality control. As regular quality control approaches, we're unable to find that this given plate is is not, let's say, faulty here, because when we were looking at the distribution of various features and intensities, they were still within given range of acceptability. But when we performed hit identification, we can see that we have some major issues here. This is the same experiment. We have the same plate layout, of course, different treatments, but there is a major problem with the anomaly score. As you can see, that there's some kind of a wave pattern, so it's most likely some kind of an experimental error where somebody, I don't know, poured something over the plate or blown some hot air on the plate or something like this, where we can see a direct effect on the cells, where still, when you look at the cells themselves, these effects are not visible with naked eye. Another thing that we can use it is for detection, for example, of well infection, where, again, regular quality control would not find this troubling, as when we average the intensities over the entire well, the effects are not significant, but our hit identification module found that this area here is anomalous, which is here is visible with naked eye, but when we have hundreds of plates with hundreds of sites per each well, then it's completely unfeasible to look at them by hand, so we have to rely on automated processes. Yes, so these are, to sum up, we can work with high content imaging to help alleviate the problem of analyzing terabytes of data, hundreds of terabytes of data in reasonable times with high accuracy. And we can then use these kind of currently developing high content screening approaches in a high throughput manner and get massive results and basically switch from targeted high throughput screening as it was done years and years up till now to phenotypic one where we can observe not only the effect on the cell and predict the target that's being hit by the given compound, but also other effects like toxicity and cell death. Yeah. So in case of challenges here, the greatest challenge, as always, when working with AI, it's data availability. So we are open for any collaborations, both scientific and commercial. If you have any imaging data within your lab, we are very happy to work with you to set up some kind of a proof of concept and then have a talk on how can we apply our methods here. Even to relate a bit to the previous ones, we even have a paper out where we were predicting strains of bacteria from cell imaging, well, infected cells. So as well as, of course, we have a lot of job openings available if anyone's interested in working with us. Yeah. So thank you very much, and you can reach me on the platform.
Great, thank you so much, Krzysztof. Thanks. So do we have Ms. Gabriela Krężel in the house? Sorry, I didn't mention that earlier, but Natalia Haraszkiewicz is not here, so we're going to move to this other presentation. So we already, so the title of this track is Pioneering Health Solutions. So we already heard about small molecule development. We heard about all the possibilities for wellness and for therapeutics that the microbiome gives us, but Pioneering Health Solutions actually goes beyond that. So now we're going to hear from Gabriela Krężel about European project focus on promotion of tools supporting vaccination. The floor is yours, please.
So good afternoon, everyone. My name is Gabriela Krężel, and today it is my pleasure to present the EUVABECO project, so European Vaccination Beyond COVID-19. And the Department of Health Promotion and eHealth at Jagiellonian University Medical College is a partner in this project. And at the beginning, it is important to outline that it's European project focused on promotion of tools supporting vaccination. But I would like to start from brief presentation of our department. So the Department of Health Promotion and eHealth is part of Institute of Public Health, which is part of Faculty of Health Sciences of the Jagiellonian University Medical College. Our main activities involve research, university teaching, and involvement in projects. Both National Science Center funded project and European funded project. And today our attention will be, of course, paid on involvement in European funded project. Our main research topics are connected, of course, with health promotion and eHealth, but we distinguished four main research topics. And first of all, determinants of health. So the department tried to identify the causes and consequences of individuals' health behaviors and commercial determinants of health. The second area is health literacy. So the department tried to identify the determinants and consequences of individual health literacy level. In the most common form, so health literacy, but also in a form of e-health literacy and food literacy, for example. The third area is use of digital health, and the department tried to identify attitudes and perception toward new technologies. So, for example, artificial intelligence, telemedicine, humanoid robots, and among society and healthcare professionals. And the last one is attitudes towards vaccination. So the area most connected with our today topic. And in this field, the departments try to identify factors contributing to anti-vaccination attitudes and frequency of vaccine hesitancy. So now we can move to our main topic, so Ryvu project. The aim of the project is the assessment of implementation of vaccination supporting tools in Europe. We are currently in 30% of advancement, so the starting date was January 2024, and the project is 30 months, so the final date is June 2026. And in this project, 14 partners from 9 countries are involved, and University of Crete act as a coordinator. And total budget is over 8 million euro, and around 80% of this is contribution from the European Commission. And the project consists of 7 work packages, and the first 2, so project management and member state uptake and the dissemination is the general, but for example, the third work packages, so map innovative practices in vaccination and select reference practices. We have already finished this working on this task, so I will show the results on the next slide. The fourth work packages is derived tools, requirements, and validation plans from reference practices. The next is develop implementation plans, including verification, and the last work packages, verify implementation plans and evaluation of pilots will be held in the last stage of the project duration. As I said, we have already finished working on work package 3, and the result of this work package are selected reference practices. In this work packages, our task was to identify the practices supporting vaccination, and we drew from different types of sources. So, for example, great literature, scientific publication, and the other guidelines. And for example, selected references practices are vaccine administration by pharmacies, enabling vaccination in mobile clinics, sending personalized messaging, using secondary data. And now we are working on implementing transformative tools in a form of pilot projects. And in Ryvu project, we have 5 transformative tools from different areas. So medical tool, social tool, industrial modeling and forecasting tool, and digital tool. But I will focus more on medical tool and modeling and forecasting tool, because we are currently implementing these tools at Jagiellonian University. So first, clinical decision support system, so medical tool. We are implementing this tool in cooperation with SIADEM. SIADEM has already implemented this system in France, and we are trying to implement this system in regards to vaccination among adult population in Poland. And so we are trying to adjust this system to our Polish conditions, so taking into consideration, for example, the reimbursement level in Poland, the Polish medical guidelines, and some other conditions. And the aim of this system is to enabling enabling informed vaccination choices. So, for example, somebody will enter the data, personal data like gender, age, or health status, and the system will deliver the information whether the vaccination is recommended for this person, or highly recommended, or there are any contradiction, for example, and the system will also display the reimbursement level. The second tool is modeling and forecasting tool, and we are working on this tool in cooperation with University of Ireland, but more information will be known in the future because now we are more involved in working on CDS, so clinical decisions support system. As I said, the project is on 30% advancement, and currently we are working on development of Polish version of decision support tool for adult population in Poland in cooperation with CIADM. We are also working on using Polish data on COVID-19 related outcomes in modeling and forecasting tool from the University of Ireland, and as an additional activity, we have also started working on systematic review with the aim to identify intervention focused on increasing the COVID-19 vaccination coverage in Poland. It is not necessary for the project, but we would like to summarize everything, and we started working on this systematic review. The project is more advanced now, so we encourage to visit our websites. So the first one is official Uwabeco website, which is dedicated for the project, but we also distinguish the sub-page on our Public Health Institute website, where you can find information about this project. And we are open to suggestions and ideas. So if you would like to, for example, cooperate with us, please contact us. Currently we are working in a group of 8 people, but we are open to any suggestion. And the best option would be to contact directly with Professor Mariusz Duplaga, because he's responsible for general management in the project, and he's also the head of the Department of Health Promotion and eHealth. Thank you very much.
Thank you so much, Gabriela. Fantastic, that was great. So already we heard about small molecule development, the microbiome. Now we heard about vaccines. So that's a lot to process and a lot to digest. So we split this track into 2 parts. So now we're gonna have a short break, a 24-minute break, and we're gonna reconvene at 3:20, 3:20, with Dominik Czaplicki, who's gonna chair the remainder of this track. So let's take a break, let's connect, let's talk more about everything that just happened.